Hi everyone. Today I'll be talking to you about the Human Genome Project, a topic I found interesting since I've learned about it in my high school biology class. First I'll be talking about what the Human Genome Project actually is. The HGP, also known as the International Human Genome Sequencing Consortium, was an international collaboration between France, Germany, Japan, and China, and the United Kingdom. The US also played a huge part in it. Scientists from 20 institutions spread out between those 6 countries came together on a 13-year project to map and understand all the genes, or genome, that make up the human being. The 5 main sites responsible for genome mapping were the Broad Institute/Whitehead Institute for Biomedical Research in Cambridge, USA, Washington University in St. Louis, Missouri, Baylor College of Medicine in Houston, Texas, the Department of Energy's Joint Genome Institute in Walnut Creek, USA, and the Wellcome Trust Sanger Institute in Cambridge, UK. The Wellcome Trust was responsible for mapping 1/3 of the human genome. So before I go too far into talking about the HGP, I want to take a few minutes to talk about DNA and genes, to help get a better understanding of what the project's goal and intentions were. DNA is found in the nucleus of every cell in the human body except red blood cells, because they don't have a nucleus. As we learned earlier in anatomy and physiology, DNA is made up of 4 nitrogen bases called adenine, thymine, cytosine, and guanine. It also has carbon sugar molecules and phosphate molecules that bind the bases together to form a nucleotide base. When 2 nucleotide bases are put together, they form the double helix shape you see in the picture I have here. When you put 3 billion of those pairs together, you get a complete set of human DNA, also known as the human genome. 3 billion is a huge number. To give an example, if you were to take and write down all 3 billion pairs, it would fill up 10,800 page books. DNA is split up into chromosomes. We get these chromosomes from our parents, 23 from our mothers and 23 from our fathers. The majority of the DNA is the same as every other person on the planet. It's what makes us all human beings. The small parts that are not the same are what make us individuals. Things like hair and eye color, facial features, height and weight are all determined by these base pairs. So I have a brief history of the Human Genome Project. In the beginning of 1988, a group of scientists, administrators, and science policy experts came together to work out a plan to establish the HGP. 2 years later, a 5-year plan outlining specific goals was published, and on October 1st of 1990, the Human Genome Project began. In September of 1994, a team of researchers published a linkage map of the human genome, successfully completing one of their goals a year ahead of schedule. In April of 1996, the National Human Genome Research Institute announced a pilot study that would determine if sequencing DNA on a large scale would be possible. In September of 1998, the heads of the HGP announced that they would finish the project a full 2 years ahead of the planned end date. In March of 1999, the pilot study launched 2 years previous was completed and then began to try and sequence all 3 billion bases of the human DNA. Then, in December of the same year, the whole of the genetic code of a chromosome was unraveled for the first time. On January 27, 2000, President Clinton gave his State of the Union address. In the address, he discusses the HGP. Here's a brief portion of what he said. In the new century, innovations in science and technology will be the key not only to the health of the environment, but to the miraculous improvements in the quality of our lives and advances in the economy. Later this year, researchers will complete the first draft of the entire human genome, the very blueprint of life. It is important for all our fellow Americans to recognize that federal tax dollars have funded much of this research, and that this and otherwise investments in science are leading to a revolution in our ability to detect, treat, and prevent diseases. For example, researchers have identified genes that cause Parkinson's, diabetes, and certain kinds of cancer. They are designing precision therapies that will block the harmful effect of these genes for good. Researchers already are using this new technique to target and destroy cells that cause breast cancer. Soon, we may be able to use it to prevent the onset of Alzheimer's. Scientists also working on an artificial retina to help many blind people to see. And listen to this, microchips that would actually directly stimulate damaged spinal cords in a way that could allow people now paralyzed to stand up and walk. In February of 2001, the HDP announced they would be offering a free multimedia kit that would be used to educate high school students and the general public. And then finally, on April 14, 2003, the IHGSC announced 2 years ahead of schedule that the Human Genome Project was successfully complete. So I want to talk a little bit about the cost of the project. The total cost of the project was substantial, coming in at a total of $2.7 billion. The total was $300 million less than originally planned because of the early completion date. After first mapping the human genome, a draft, or initial sequencing, was created from April 99 to June 2000. The draft cost this project $300 million to achieve. Once the draft was completed, the project was able to complete a final sequencing of the human genome. This cost the project another $150 million. With the draft and the final sequencing only totaling $450 million, you might be asking, where did the other $2.35 billion of the project cost go to? The other $2.35 billion was split between two categories: production and non-production expenses. Most of their production costs went to labor, administration, management, utilities, reagents, and consumables were among the biggest expenses in the production category. The second highest expense in the production category was the large equipment and all the sequencing instruments required to complete the project. The non-production costs went to quality assessment, technology development, the development of bioinformatics, informatics equipment, and the management of individual sequencing projects. So here I have a graph showing the cost per raw megabase of DNA sequence from 2001 to 2020. One megabase is equal to 1 million base pairs. As you can see, the initial cost of sequencing of 1 million of the 3 billion pairs in the human genome was roughly $10 million. But now, in 2020, the cost to sequence a megabase is below $10. On this slide, I have a graph of what it costs to sequence the entire human genome. As with megabase sequencing, the initial cost was very high, totaling $150 million. Since the implementation of second-generation sequencing technologies in 2008, the cost to sequence a human genome in 2020 has dropped significantly, roughly totaling about $3,000. So the results and impact of the project. After the completion of the project in 2003, the results were deemed to be an overwhelming success. The finished sequence covers 99% of the genome-containing regions and was sequenced to 99.9% accuracy. The completion of the project also led to other amazing advances in sciences. Advances such as a draft sequence for a rat genome, the identification of 3 million human genetic variations called single nucleotide polymorphisms, and the full-length complementary DNAs for more than 70% of known human and mouse genes. The HGP has made a large impact on the fields of medicine, law and social sciences, and economics. In the medical field, the International HapMap project was formed. The IHMP is comprised of 5 countries from the continents of Africa, Asia, Europe, and North America. The purpose of the project is to identify and catalog genetic similarities and differences of individuals from the 4 major populations of these continents. Another major impact the HGP has had on the medical field is the ability to identify a variety of disease-causing genes in the human population. This has led to an increased accuracy of diagnosis and has helped determine the genetic influences on development or outcomes of rare and common diseases. In law and social sciences, the finished human genome has helped the field of forensics by using a system known as CODIS. CODIS is the Combined DNA Index System. This system has helped to positively identify criminals from samples of saliva, hair, and spots of dried blood. The economic impact of the HGP has been huge. The HGP produced 3.8 million job years of employment, or 1 job year for each $1,000 invested. Personal income over the project's time frame was upwards of $244 million. In 2010, genomics was responsible for 488,000 jobs, which added $67 billion to the US economy. In the same year, industries related to genomics created $3.7 billion in US tax revenue. So here I have a video of President Clinton and Prime Minister Tony Blair talking about the HGP after it was completed.
After all, when Galileo discovered we are here to celebrate the completion of the first survey of the entire human genome. Without a doubt, this is the most important, most wondrous map ever produced by humankind. Have the great strengths and insights of the public and private sectors. Today's announcement represents more than just an epoch-making triumph of science and reason. After all, when Galileo discovered he could use the tools of mathematics and mechanics to understand the motion of celestial bodies, he felt, in the words of one eminent researcher, that he had learned the language in which God created the universe. Today, we are learning the language in which God created life. We are gaining ever more awe for the complexity, the beauty, the wonder of God's most divine and sacred gift. With this profound new knowledge, humankind is on the verge of gaining immense new power to heal. Genome science will have a real impact on all our lives, and even more, on the lives of our children. It's the full spectrum of science and technology. It is my great pleasure to turn to my friend, Prime Minister Tony Blair, who is joined in the State Dining Room at 10 Downing Street by Dr. Fred Sanger and other world-renowned scientists.
I think of my little boy, Leo, growing up and learning and knowing things that his grandfather, after whom he was named, could not even have dreamt of. When you contemplate that, it's almost not like different generations, but different eras of human existence. And there are enormous possibilities for him and his generation, but also some dangers. And our job is to try and develop the possibilities and thwart the dangers. Process of peace in Northern Ireland. And as we continue progress with another important step forward today. But with the power of this discovery comes, of course, the responsibility to use it wisely. As with the greatest scientific achievements, the ethical and the moral questions raised by this astonishing breakthrough are profound. We, all of us, share a duty to ensure that the common property of the human genome is used freely for the common good of the whole human race. The fact that we do indeed, as we've often said together, live in a global community. The first great technological triumph of the 21st century.
And this is my reference page. So thank you for watching my presentation today. I hope it was informative and that you all learned something.